Great, and we'll get started with our next presenting company, Fibrocell. Here to present for Fibrocell is John Maslowski, who's recently been promoted as the CEO of the company. And Fibrocell is an autologous fibroblast cell therapy company. The company is working on now two really proprietary or internal gene therapy programs and has partnership with Intrexon. So we'll get into some of these details. John, maybe you can start us out by describing Fibrocell's core technology.
Sure, thanks, Ted. I appreciate it. And thank you, ARM, for having us today. We really appreciate being able to come out and talk about our programs. And as a public company, I will be making some forward-looking statements. And I'll just leave this slide on. It'll give a little summary of our platform here. So Fibrocell, as Ted just mentioned, we have what we are hoping to develop out of this is a platform technology of autologous fibroblasts, so we can deliver genes of interest to skin and connective tissue. So we're primarily targeting rare pediatric diseases, obviously ones that are monogenic in nature, and I'll speak about those as we move along. Primarily recessive dystrophic epidermolysis bullosa, and the technology is essentially expansion of autologous fibroblasts, and early in passage we use lentivirus to introduce genes of interest. In our main candidate, it's COL7A1, which encodes a protein called collagen 7. There's a lot of reasons why we chose lentivirus, mainly because of its integrative nature and that this is a chronic disorder, and also that the gene inserts are quite large, up to 9 kb, so we really did need the packaging capacity for that. So, what we hope to do is continue, show proof of concept in this first one, which we are currently in phase 1, treated our first patient just in the end of February, and then move on into other indications. As you can see here, linear scleroderma, which is an autoimmune skin disorder, and we also have some interest in larger indications such as arthritis and joints. That's early research. Really do need to establish the platform first.
Absolutely. And perhaps you can start off the first program, as you mentioned, is FCX007 for RDEB. Maybe you can tell us a little bit about this horrible condition and really how affecting collagen 7 may be able to help.
Sure, and I'll just throw a slide up that talks a little bit about the disease. Oh, I think it's not in this particular deck. I apologize. So RDEB is generally pediatric in nature because of its mortality rate. It's a single mutation in a protein, as I mentioned, called collagen 7. And what occurs is collagen 7 makes up a structure called an anchoring fibril, which binds the layers of skin together, so the epidermis to the dermis. And think of it like Velcro, sort of like you have on the top, you have like a hook, and then your ECM proteins thread through that hook from underneath in the dermis, and then that tightening of that keeps your skin together. Unfortunately, in these kids, they lack this protein, or they form non-functional collagen 7, so their layers of skin essentially float on top of one another, and as soon as they have just the slightest bit of lateral friction, they get blisters, and then they spread, and then it becomes a chronic issue with wounding. The main, there are no approved therapies at the moment. Right now, it's really wound management and care, antiseptics. These sorts of things are really the state of the art. They do a lot of surgeries for esophageal dilatation and for finger separation surgery because they tend to bond with all the wounding and the healing. A lot of that work cost right now is coming out of the pockets of the patient's families. Very little reimbursement on the supplies right now. So there's a huge economic burden. And really the mortality, usually once they reach their late 20s and their 30s, comes from mainly squamous cell carcinoma with all the turnover of the cells and in the skin, and then obviously sepsis. Malnutrition used to be a large issue, but they typically fit them early with feeding tubes. So it's a terrible disease to live with through their lives. Obviously it affects their families, huge psychological impacts. And so that's why we hope we can make a big difference here.
Yep, absolutely. I think you're doing really important work here. Now, if I'm not mistaken, you have begun dosing patients. I think the first patient, which is a great milestone, I'm really, really pulling for you guys here. Maybe you can tell us about the phase 1 trial design and when we could get data.
Sure, absolutely. So we've designed our initial trials at phase 1/2. It's targeting to enroll 6 adults in phase 1, and then in the phase 2 portion, we have an additional 6 patients, and our goal is to include pediatric portions into that second phase of the study. It's primary, we're looking at safety, but we're also looking at some mechanism of action endpoints. In this disease, there's actually quite a lot of academic research that's been done, and since we know the protein is collagen 7, looking at biopsies, cross-sections, looking at immunofluorescence with an anti-human Col7A1 antibody, and even electron microscopy can actually physically view the morphology of the anchoring fibrils. We'll be looking at that as well through the progression of this trial, and then also using 3D imaging to look at wound healing rates and also looking at wound durability from those as well, from 4 weeks out to 52 weeks with a number of endpoints. As Ted just mentioned, we did just dose our first patient at the end of February. Our protocol design requires us after 4 weeks to have a DSMB look at those data and then ensure there's just generally safety since it's a new gene therapy candidate. We're going through that process now, and then from there, we'd be released to then treat additional adults. So we're hoping that's our next 2 short-term milestones.
And John, I should just ask, what is the dosing of these patients? And beyond just dose, so again, we're talking essentially about modified autologous fibroblasts that have been conditioned to express collagen 7. So is this given topically? Is this given intravenously? How do the cells actually port to the sites of injury and express the collagen?
Yeah, it's a great question, and well, I'll explain over this because they are actually intradermally injected at the site of lesions or at the target site that we plan to treat. And probably first thing we're all thinking to ourselves, well, it's an autosomal recessive disorder. How do you plan to treat the body? Now, unfortunately, trying to do this intravenously, the protein is large. There's a lot of problems that could potentially emerge from that. There's some early protein data that may suggest that an intravenous delivery of this could actually be problematic from a toxicology standpoint. So we feel that the localized version, and also in skin, as compared to doing this in some other gene therapy programs, gives us somewhat of a safety advantage. Has the safety component that we need. It allows the cells to actually be implanted at the site of dose, a site where the actual disease is manifested, so we can produce that. So our goal is to treat wounds to a certain size over the course of their lifetime. This isn't like an AAV treatment. We can have redosing as a possibility here because they're ex vivo modified. What our physicians say is we'll treat trouble areas like joints, and then eventually we'll go, next time they come in for other surgeries, we'll do other parts of the body, and then we'll continue, and that'll be their course of treatment over a lifetime.
And from the preclinical data, what is your kind of current thinking about durability? Are these cells engrafting, since they are autologous fibroblasts? I mean, they're actually very proficient producers of proteins. So I would think that from a sheer, you've picked the right cell to actually express these proteins, but, how long do you think you may actually get well?
Yeah, and our preclinical data is not out quite far because, obviously we need to get to IND at some point. We do have toxicology data that goes out 6 months. We have some pharmacology data that's a bit shorter than that. I can give some, just some hypotheses though, and we designed our trial actually to see this durability goes out 52 weeks. We're also doing some additional DNA, mRNA analysis to see what's going on in the skin, but primarily, looking at these mechanism action and photography will be our endpoints. But the cells obviously are autologous. There's been some published literature on how that works out. The protein itself, is actually it's a high tensile strength protein. We hope the cells produce that, and then that protein can have some longevity on its own. So there's a lot of hypothetical reasons why you should do that. Obviously, we need to show that clinically, and one of our goals is to show, that we can that we get longevity of the protein. But also, one of the benefits of being able to intradermally inject this is we can treat other areas, we can retreat areas. We obviously don't want to be doing that all the time. That's what we're trying to find out on this longevity.
Understood. And we've talked a lot about some of the changes that have taken place in the regulatory landscape. How could 21st century cures help you guys? Obviously, it's going to be data dependent, but where do you see that as potentially shortening the path?
Yeah, it's a good question, and obviously a discussion we can have for a full conference here. But for an early phase asset, being in phase 1, having earlier assets, if you look at the Cures Act, there's a lot of sections that are now helping us to define really, what are the right standards for not only rare diseases, but also for regenerative medicine. Obviously, it was the big part of the Cures Act, and now we have RAD designations, and I think that some of those, including accelerated pathways, will become more and more available for companies who are starting off and now collecting some early data. I think the patient experience component of it could be interesting for rare disease. The endpoints aren't quite as designed as large diseases, and I think that, as FDA agree, will take more and more into account, will be very helpful. And of course, we have, if you see here, we have pediatric rare disease designation for this candidate. The Cures Act also extended that out to 2020 with options out to 2022. So clearly, even from a corporate standpoint, that'll be very helpful for us.
And we mentioned Intrexon. Tell us what they're doing to help you guys with the technology.
Yeah, so the history of that relationship came from us, and back in 2011, we had approved a cell therapy called Lviv for an aesthetic purpose for facial wrinkles. As the time went on and we started shifting towards rare diseases, what we found out talking with Intrexon is there a way to deliver these genes locally? And what they brought to the table was a very rapid development of vector cassettes that we can actually screen, look at protein expression, and narrow down to what would be the right for our vector, and then look at different vector classes. We looked at non-viral, viral, chemical, electroporation. With a big gene, unfortunately, there's some limitations of what you can do. And eventually, we were able to narrow that down using Intrexon to get ourselves to a candidate, which we scaled up GMP and are using now in the clinic, and it works quite well for us.
Great. Well, I really, truly wish you all the best on that program.
Hey.
I think you're doing important work there. I want to spend just a quick second on FCX013 for lineal scleroderma. Maybe mention what this condition is and how you're going after treating it.
Sure. So our next candidate is a preclinical candidate we're looking at right now called linear scleroderma. This is an autoimmune reaction that happens. Generally, the linear type occurs in children, usually between the ages of about 15 to 15, where you get these sort of linear, kind of like ropey sort of scars on your face. Scar lesions, you get this sort of thickening of the skin is really what is happening, and that's basically the dermal layer is becoming thick, and it's because of an excess production of collagen 1 and 3. And what we're trying to do here, sorry, just to finish on that, it's very debilitating and cause developmental issues and cause range of motion issues. If it's done across the head, it can cause neurological disorders. And right now, the standard of care is corticosteroids and light therapy and some other things. So we think this is a great candidate to help patients. And we've developed 013 to be, it's a gene that instead of creating collagen, it actually is an interstitial collagenase. And then we've also incorporated Intrexon's Rio switch control, which gives us some ability to dial the protein because you don't want an interstitial collagenase to be functioning on its own in the body and being pumped out. So that's in a preclinical model now. It's a bleomycin model. We did a very large non-GLP study. The goal is to do dose ranging because you have the ligand, you have cells, you have vector, copy number. There's a lot of things we really need to analyze to make sure we have the right dose. So we're there now.
And again, since this is an investor conference, you recently were able to raise some capital. How much is Fibrocell currently holding cash, and how long will that fund the company?
Right, yep. And at the end of 2016, we had $17.5 million in cash. We recently just raised $8 million with some of our insiders. And that money will last us into the second quarter of 2018, which does get us past the inflection point for 007, which is we hope to report data on that in the third quarter of 2017, and then obviously have some burn rate left over for that as well. So we are funded through our data point. We think we have a good handle on the trial. We have a good team intact, and looking really forward to helping these patients.
Good. Well, wishing you all the best on that. Love to be helpful in any way I can.
Of course. Thanks,
Thank you
Ted.
very much, Sean.
All right, thank you. Thanks, Ted. I appreciate it.